RT-PCR RNA quantification. Overview. RT-PCR RNA quantification will be considered under the following headings: Introduction, Terminology, Spectrofluorometer, TaqMan Probe, How does it work, Amplification Curve, CT value, and Sample report. Introduction. RT-PCR test involves 4 steps: sample collection, RNA extraction, RNA amplification, and RNA quantification. In the previous video, we discussed about RNA amplification. In this video, we will be reviewing the process of RNA quantification. Terminology. Amplicon in PCR refers to the DNA strands that are the product of amplification. Quantitative real-time PCR can be used to quantify the amplicon in real time. This term is frequently confused with reverse transcriptase PCR or RT-PCR. Spectrofluorometry is a technique to detect and analyze the fluorescence in the sample. Quantitative real-time PCR is a combination between PCR and spectrofluorometry. Fluorescence is the phenomenon in which a material known as fluorophore emits light of lower energy and longer wavelength when it is illuminated by another light of higher energy and shorter wavelength. Spectrofluorometer. Spectrofluorometer is an instrument that analyzes fluorescence from a sample. Components of instrument are light source, excitation monochromator. It allows only the radiations which excites the fluorophore in the sample to pass through. Sample with fluorophore. Emission monochromator. It allows only the radiations which are emitted by the fluorophore in the sample to pass through. It is always kept at 90 degrees from the incident light. This minimizes the chances of transmitted or reflected light reaching the detector. Detector. It converts light energy coming through the emission monochromator into electrical signal. Charge coupled device or CCD camera is commonly used as a detector in spectrofluorometry. From detector, the electrical signals are passed to the readout device, which displays in the form of graph. TaqMan Probe. TaqMan Probe is a DNA probe which consists of a single-stranded DNA coupled with 2 molecules. There is a fluorophore at the 5' end and a quencher at the 3' end. Quencher is the inhibitor of the fluorophore by absorbing any fluorescence coming out of the fluorophore. As long as the fluorophore and quenchers are placed close to each other, we cannot see any fluorescence coming out of the fluorophore. If they are separated from each other, then quencher is no longer capable of absorbing fluorescence from the fluorophore. With each cycle of PCR, more fluorophore molecules are released, resulting in an increase in the fluorescence intensity proportional to the amount of amplicon synthesized. This allows quantification of the amplicon. How does it work? As discussed earlier, after the first step of thermal cycle, that is denaturation, the 2 DNA strands separate from each other. Now in the second step of thermal cycle, that is annealing, the primers will bind to the 3' end of DNA sequence of interest. This is when the TaqMan probe also recognizes and binds to the DNA sequence of interest. In the third step, or extension, the DNA polymerase will start copying the DNA sequence with the help of DNA content or dNTPs. As it reaches the probe, it will start to degrade the probe and release the fluorophore. Once the fluorophore is released, it is no longer close to the quencher, so quencher cannot absorb the fluorescence and it becomes detectable. Polymerase in the meantime will continue to copy and degrade the probe. Finally, the probe will be completely degraded and its components, including quencher, released. Amplification curve. After each thermal cycle, the number of DNA sequences of interest double. As the PCR progresses, more and more DNA would be produced, more TaqMan probe will bind to DNA sequence of interest, and more fluorophore will be released. This in turn can be detected by the spectrofluorometer. This way, there will be a progressive increase in the detectable fluorescence, which will be in direct proportion to the amount of replication of DNA sequence of interest that has taken place. This is represented by an amplification curve. On the y-axis, we can see the fluorescence. On the x-axis are the cycles of PCR. CT value. The amplification curve can be divided into three phases: early phase, exponential phase, and plateau phase. During early phase, the reaction is just beginning and very small levels of fluorescence is generated. A threshold value is the point at which fluorescence reaches values above the baseline levels. In the exponential phase, the successive cycles of amplification results in exponential increase of copies of the target DNA, leading to accumulation of fluorescence. In the plateau phase, amplification is no longer occurring exponentially since reagents have been used and fluorescence levels do not increase. CT value is also known as cycle threshold. It is the cycle at which the amplification curve intercepts the threshold line. Let us compare an amplification curve 1 to amplification curve 2. The amplification curve 1 suggests that at the beginning of PCR there was higher amount of DNA present, so that with amplification the fluorescence started increasing exponentially. The amplification curve 2 suggests that at the beginning of PCR there was lesser amount of DNA present, so it took several cycles of PCR before the fluorescence started increasing exponentially. Therefore, the sample represented by curve 1 has lower CT value than the sample represented by curve 2. In other words, the CT values are inversely proportional to the amount of DNA in the sample. That is, the lower the CT value, the greater the amount of DNA in the sample. Sample report. This is sample report of a patient suffering from COVID-19 infection. As we can see here, the sample type is nasopharyngeal swab. Methodology is quantitative real-time polymerase chain reaction or Rtpcr. Target genes include ORF1ab and RDRP gene. CT value is 14.438 and result is positive. Thank you.